This is presented by my co-principal investigator, Luciano Costa. This was a really, I think, an innovative and ambitious trial where we combined the baseline or the platform or the backbone of Mezigdomide dexamethasone with a variety of novel agents. And really, the important focus was on Tazemetostat, or Taz for short, and on Trametinib. I'm going to really focus on those. There's obviously a strong preclinical rationale for Tazemetostat in combination with Mezigdomide and Dex, given its effects in terms of some downstream targeting in combination with cerebral and E3 ligase modulation and Icaros and area loss degradation. I think the rationale for Trametinib is similarly strong, but suffice to say the clinical data, which Luciano presented really beautifully, were as follows. They showed that essentially you could safely combine Mezigdomide and dexamethasone with both Taz and Trametinib. The combination with Taz was clearly effective. With Taz was clearly effective, and in that context, we were able to see a response rate of around 60% from the combination in heavily pretreated patients who had also had prior BCMA and CAR T therapies. So, a very important unmet medical need. What was really interesting, with my view, was the Trametinib data. There, we showed that the combination of Mezigdomide and dexamethasone with Trametinib in a substantial cohort of patients, the total overall number of patients was approximately 60, and then if you looked at the different cohorts, there were about 22 each, right? And there were 3 cohorts. The third cohort was with a different inhibitor that probably is not showing the same efficacy. This was an experimental drug targeting bromodomain inhibition, and that particular cohort didn't really meet our expectations. Safety-wise was fine, but the efficacy signal really wasn't particularly strong. So on that basis, really Taz and the Trametinib data stood out. So as I mentioned, the Trametinib data were noteworthy for the fact that in a heavily pretreated population, including patients who'd had prior BCMA exposure, be it antibody-drug conjugate, T-cell redirection, or CAR T therapy, we saw a response rate in excess of 70%. In fact, it was 75%. And if you looked at certain cohorts, it was over 80%. So we were really encouraged by this with a manageable safety profile. So we have to be careful because it was preliminary data, but nonetheless, the fact that we're seeing such a responsiveness from an all-oral regimen in such heavily pretreated patients who were not only triple-class refractory, but also refractory to BCMA targeting approaches was, in our opinion, particularly noteworthy.